And a few years ago we have broken through this barrier, so we can now study these systems routinely. And here are some benchmarks that give you an indication on the performance of such simulations. So this is for a network of 100,000 neurons and 1 second of biological time, and for different computers that we played with in the past. And you see that here you can, by distributing the network over several machines, you can reduce the simulation time from half an hour to a few seconds. Now, this may make you wondering if this is really worth the effort. If I can have the time to wait for half a minute, maybe I can also wait for half an hour and do some useful analytical calculations in between. But the problem here is that this is for a static network, but what we are really interested in are plastic networks, and if you study plasticity or even structural plasticity in the network, then you are concerned with the biological time scale of minutes, meaning that this would convert to hours of computation time. And what we are usually trying to achieve in the lab, that we can do one simulation in 24 hours, so that in one day you start a simulation and the next day you have the results. So what this technology enables you is to study large-scale plastic networks. But what we also observed is that for simpler systems like this here, which is a static network, that people started to work with the system in a qualitatively new way, because now these simulations are quasi-interactive. So you can change your parameter, press the return button, and basically immediately get back the results, meaning that you now can play with such simulations like you would play with a dynamical system with Python or in the past with MATLAB.
One question.
Yes.
The outcome of the simulation is just the final rate, or you want to compute something more like correlations or something like that?
Of course. Eventually, we also want to study correlation, but this is only meaningful if the firing rates are in the right regime. So this is just the first question to ask. Right. So this is our computer in the lab. So from the front, it looks like a standard computer cluster, but if you look from the back, you see all these quite wild-looking cables coming out, and so the black ones are connections of a specific communication interface, where the switch is located here in the middle, and this is used for transmitting the spike data from one box to each other. And the difference to the standard computer network is not so much the bandwidth, but that the latency is much shorter than with gigabit Ethernet. And just to demonstrate to you that nowadays it's quite easy to assemble this computer power in the lab, is that with the system we have here, if I do just a very rough calculation, like number of computer cores times speed of the processors, then I end up with this figure, which is about 70% of one rack of the last version of the Blue Gene supercomputer. So we are already in the range of supercomputing here. And this is a simulation result on such one rack of a supercomputer, where we use up to 1,000 processors here, and you see that also here the scaling of the simulation is quite nice. Now, and just to mention that of course we are not doing this software development alone, but in a collaboration of several labs, which is called the NEST initiative. where the Honda Research Institute is an important partner of us. They are investing a lot in this software development, and by now we are using this teaching also in some international courses. And I should add that that with this, the main goal of this national initiative is not only to produce this software, but a maybe more important goal is to systematically publish the simulation technology that is useful for doing this kind of research. So now back to neuroscience. This is the first result that you can get out of such a simulation. So this is time. Here the individual layers are indicated, and blue is the spiking activity of the excitatory neurons in a layer. Red is the spiking activity of the inhibitory neurons in a layer. And what you immediately see is that the spiking activity is different between excitatory neurons and inhibitory neurons, and the spiking activity is different between the different layers. And it looks quite irregular. And now, this is now already an interesting result, because remember that the dynamics of the excitatory and inhibitory neurons was exactly the same. And anyway, we get out that the spike rate of the inhibitory neurons is higher, and also the properties of the neurons is exactly identical in the different layers, but still we get different spike rates. So all the heterogeneity we see here is a reflection of the structure of the system, not of the dynamics of the individual components. And another point is that now it's possible by the so-called laminar electrodes to determine the spike rates in the different layers, so we can really check for this data. And I'm mentioning this study here because it showed that in layer 2-3 we really have this amazingly low spike rates as are shown here. Now, what about the role of target specificity? The results I've shown you so far are for this, or best estimate of this value of the target specificity. But because we have a model, we can now start to play with these parameters. And what I will do in the following slides is that I will reduce the target specificity of these feedback connections in this direction, so I will make them more positive. And here on the lower left, you will see how the activity changes. So you see that we get more and more spikes, and also you see that some oscillatory structure is developing, and here you don't see anything anymore. But now I zoom in, and you see that now the whole thing is epileptic and synchronously firing. So already from this we can guess that this negative target specificity has a role in stabilizing the system. But this is still for this rather boring stationary situation where there's only DC input. And here this expressed more quantitatively, where you see that with increasing target specificity, although this is logarithmically scaled already here, we see this sharp transient in the spike rates eventually. Now here is a more interesting situation. Here we applied a pulse of activity like it would come from thalamus to layer 4 and to layer 6. On the left here is the healthy situation, and we can observe that a pulse of activity is nicely propagating upwards to layer 2-3 and then down to layer 5. And